Today we're going to talk about what happens if you only have a little bit of DNA to work with from your crime scene. So we've already talked about how to extract DNA out of the cells, and we went through that whole process. But what happens if you only have a minute, tiny little bit of DNA that you found at your crime scene that you were able to extract out? What do you do to get a sample big enough for profiling? So you do the polymerase chain reaction, or we abbreviate it PCR, so oftentimes we just say PCR. It's a reaction that allows for trace amounts of DNA to be turned into large quantities of DNA so that multiple tests can be done on it. It can be run multiple times so that you're getting the most accurate results possible. The polymerase chain reaction is extremely fast. Okay, they can make large quantities, like billions of quantities of the same DNA in about 1 to 2 hours. They use it for forensic testing or genetic engineering, depending on what they're looking to use it for. So let's take a look at the process. Okay, on your notes you've got 4 boxes for the 4 steps. I highly recommend that you write what the step is and that you draw a picture of what's happening in each step. And we'll kind of go through the process a few times. So step 1, you have to heat the DNA sample up to about 98 degrees Celsius for 5 minutes to denature or separate the DNA strands. So it's kind of like replication, where you have to pull apart the DNA. But instead of there being an enzyme to do that, they use heat to do that. It denatures it and splits it apart. So the very first step is going to be to split apart the DNA into 2 separate strands. So you have a top strand and a bottom strand. The second step that they do is they take the sample and they cool it back down to about 60 degrees, and they add what's called a primer to each strand. A primer is a short strand of DNA. It often matches what's called the flanking sequence, which is the location on the DNA that they actually want to replicate. Because they don't usually replicate all of the DNA, they usually replicate sections that they want to actually test. So they add a primer, which is a short strand of DNA that provides a start sequence for this process. And if you're taking a look over here, this is a primer, and this is a primer. Okay? So they cool the sample down and they add primers to each strand, which is a short strand. It'll be the opposite of, the primer will be the opposite of what the strand is. So, like if right here was A, T, T, the primer that they would add to it would be TTAA. It would be an opposite sequence. Okay? So they cool it down, they add the primers, which are short strands of DNA that provide a start sequence. Then they also add free-floating nucleotides and DNA polymerase. Okay? Remember, the DNA polymerase is what goes through and kind of binds everything back together. So they'll add nucleotides, your A, T, C's, and G's, and then they'll add the DNA polymerase. So here's your DNA polymerase, here's your free-floating nucleotides. Those get added, and then they heat it back up to about 72 degrees. Just a little bit warmer to make the process happen faster. The DNA polymerase goes through and binds to the primers. That's why you added them, so the DNA polymerase would have somewhere to attach to. And then it goes through and it synthesizes the complementary strands of DNA. So it goes through, and if there's a T, they add an A. If there's a C, they add a G, back and forth. And then it adds the backbone so that you're getting 2 complementary strands of DNA. So when you finish one PCR process, you have 2 identical strands of DNA. 1, 2, each with an old strand and a new strand in it. So basically what PCR does is it totally replicates in a test tube the process of DNA replication. Okay? Scientists learned how to basically modify the steps so that it would happen in a basically test tube setting. There's a machine, okay, that they put like kind of all the supplies into, and then it changes. It's called a thermocycler. It changes the temperatures back and forth, and that's what kind of starts and stops each part of the reaction. So first it heats it up to 98 degrees. Okay, that separates all the DNA strands that they're looking for. It cools it back down. They add the primers, okay, which are the short strands of DNA that are going to provide the start sequences for the locations that they want to replicate. Then they add the free-floating nucleotides and the DNA polymerase. They heat it back up to 72 degrees, and that causes the DNA polymerase to bind and synthesize the complementary strand of DNA. So when they finish after one cycle, they have 2 copies. So let's take a look at this a little bit more in depth. Kind of give you an idea of what it looks like. Okay, so they're going to warm it up. They're going to put the DNA that they want to replicate into a thermocycler. Okay, they're going to warm it up. Here's your 94 to 96 degrees, 98 degrees, depending on where it wants to be. Okay, for several minutes, and that's going to denature. So you can see it split it, top and bottom strand. They lower the temperature down between 50 and 65 degrees Celsius. They add the primers. Okay, and so there's your primers. Okay, so I've got one primer on the bottom strand, one primer on the top strand, and that's going to be complementary to what's called the flanking sequence, which is going to be a location on the DNA that they want to actually copy. And then notice, now they're going to heat it back up right here to 72 degrees, which is where the DNA polymerase is going to come in, and it's going to go through and bond the nucleotides together, and now you have 2 new strands. Then they're going to do the same thing. Okay, they're going to warm it back up to your higher temperature, 94 to 96 degrees. It's going to split the DNA. This time it's going to split a top bottom, a top bottom. They're going to add primers. Okay, so you're going to have primers again. Then they're going to put it at 72 degrees. They're going to add DNA polymerase. Now, after your second cycle, you have 1, 2, 3, 4. Then they're going to do a third cycle, but this time in the third cycle, they'll replicate all of these. So the way that the PCR process works efficiently is because every time you copy everything that you made in the previous step, and so it exponentially grows the amount of DNA. So here's your third cycle. They're going to denature the 4 that they made. Okay, so now you're going to technically make 8. 1, 2, 3, 4, 5, 6, 7, 8. They'll add the primers, add the DNA polymerase, extend. Now they have 8. Now for the 4th cycle, they'll take all 8 of these and denature them, and it'll keep exponentially growing. Okay, so now we have 16, and then we have 32, so on and so forth. So that's the beauty of the polymerase chain reaction, is that once you start getting high numbers of replicated DNA, you're quickly making hundreds and hundreds and hundreds of copies of it, because once you get a little bit higher up in the process. So you can see here, okay, here's your first cycle. DNA copies 8, DNA copies 16, 64, okay, and pretty soon what happens is they're only targeting certain areas of your DNA. They're not doing all of the DNA, they're only usually doing the specific sections that they want to look at and compare with the crime scene. But after about the 8th or 9th cycle, now they're only getting those target copies. They're not getting all the DNA anymore. And you start to get, you can see here, right? 16,000 copies, 32,000, 65,000, and we're only on cycle 18. Okay, so if they just keep infinitely going, right, you can see we get to extremely high numbers by like the 30th cycle. So in 1 to 2 hours, we can have, right, close to 1 billion copies of whatever specific DNA that we're looking at through the polymerase chain reaction. So the good thing about it is we can quickly take small amounts of DNA, even if we only have a small sample, we only found 1 small blood drop, etc., and we can quickly replicate what we're looking for so that we're able to run a DNA profile. couple of issues with the polymerase chain reaction is that if for some reason your original DNA sample is contaminated in any way and you run it through PCR, you have all the contaminated DNA in here. So if somebody wasn't wearing gloves, if for some reason the person who was running the PCR reaction, some of their DNA got in the sample for whatever reason, there's no way to re-separate it out. So it's just a completely contaminated cycle of it. If that was the only kind that you had, that would be a big problem. So there are a few issues with it that they have to be extra careful with. But overall, PCR has drastically increased our ability to DNA profile because it allows us to take those small amounts of DNA and make enough to actually work with.